My name is Rita, and my work is about laser-induced graphene-based platform for dual recognition of molecules. This is divided in four parts: introduction, experimental and methods, results and discussion, and some conclusions. Why use the commercial screen-printed electrodes have many analytical advantages, such as ability to measure small currents, rapid response to changes, and apply potential, low ohmic reduction. However, sometimes these devices require a complex process, rendering expensive and time-consuming production procedures. As an alternative, we have the laser-induced graphene technology, and show several advantages. For example, high electrical conductivity, high resolution, an expensive and high throughput solution, and several applications from biosensors, UV sensors to capacitors. This technology is based on laser radiation of polyamide substrate that photothermally convert into 3D graphene porous structures. The novelty of this work is to announce and demonstrate the freedom of the design that the laser-induced technique provides, a dual sensing platform combining two electrical chemical cells. In the same platform was tailored for detection of amoxicillin and ascorbic acid using a molecularly imprinting polymer technology. So we have here a substrate from graphene, and then we put a suitable layer, can be a conductive polymer. After that, we use electropolymerization technique and bulk in a template and monomer together, forming a film. And finally, the last step was remove the template, maintaining the integrity of the polymer. So, the amoxicillin belongs to penicillin class against to gram-negative and positive bacteria, and is the most penicillin applied in aquaculture. Ascorbic acid is known as vitamin C and plays an important role in diet of fish. Also acts against to gram-negative and positive bacteria, and is an important micronutrient for normal growth and physiological function of most aquatic animals. So, it is important to have an analytical method capable of monitoring both analyte and water. Here, it's possible to see the several steps for assembly for each analyte using a molecularly imprinting polymer. So, for amoxicillin, you use a conductive polymer. In this case, it's a P dot. After that, we mine the surface with a 4-aminophenol, and then we'll have to polymerize with amoxicillin and EBT as a monomer. EBT is a recurrent black tea, and then the final step was remove the template, maintaining the integrity of the polymer. For ascorbic acid, you use the same technology, electropolymerization, with a pyrrole as a monomer, a different monomer than use the MIP for amoxicillin. And the final step was the same thing, remove the template, maintaining the integrity of the polymer. So, to follow each modification on top of the surface, we use two electrochemical measurements, cycle photometry and scale wave photometry. The first test was testing reproducibility of several sensors, different sensors, to use the same assembly to follow. Before cleaning, after cleaning, and after the electrode position of P dot. And it's possible to see in both electrochemical measurements small errors in different types of sensors. This is very good, is reproducible sensor. Also, we tested analytical performance of the sensor using amoxicillin as a proof of concept. First, when we increased the concentration of the molecule, in this case amoxicillin, decreased the current, and it's possible to see the linear range between 50 nanomolar and 0.9 millimolar, with a good square correlation coefficient and good limit of detection down to the nanomole level, 11.98 nanomolar. As a negative control, we use the same technology, molecular imprinted polymer using the same polymer, Ericon Black chip. However, we use without the template, and it's possible to see when we increase the concentration of amoxicillin, we have a random response ups and downs the current. Also, we test the same thing with real samples. We spike the water from well with two analytes, ascorbic acid and amoxicillin. When we increase the concentration of these both analytes, increase the current in water from well. And it's possible to see a linear range between 1 nanomolar and 3.5 millimolar with a good correlation coefficient. Combine the laser-induced technique and molecular imprinting technology to produce a dual sensing platform for detection of two different molecules, in this case ascorbic acid and amoxicillin. These sensors are highly reproducible, low cost, made a flexible material and produced by a technique that allows the freedom of design with high resolutions. Dual MIP devices are also able to successfully monitor both binary standard solution and binary sample from environmental from a well. This technology is a promising method because in the future on-site analysis is possible to detect different analyses and can be adapted in different areas, because we use this in this case for culture, but it can be adapted for health to detect two different cancer biomarkers or other biomarkers or else. Knowledge for my supervisors, Professor Elvira Fortunato, Professor Goretti Salas, and my co-worker of this work, Ana Carolina Marques. Thank you very much for your attention.